Thank you for watching this presentation. My name is John Lambert, and I'm the CEO of Amplia Therapeutics. This presentation was one that I gave at the company's annual general meeting held in Melbourne on the 25th of August 2022. If you please bear with me for a second while I share my screen, I shall take you through the presentation again. Please also note the company's disclosure statements shown on this slide. What I'd like to do during this presentation is take you through a bit of an overview of Amplia Therapeutics and our year in review. For those of you who are less familiar with our technology, I'd like to just spend a few minutes talking about that. But most of this presentation is devoted to describing the ACCENT clinical trial, which is a phase 1b/2a clinical trial recently initiated by the company in patients with first-line patients with pancreatic cancer. At the end of the presentation, I'll touch on our plans for the coming financial year. I think it's important at the start of the presentation like this and on the occasion of an annual general meeting to talk to our shareholders about the values that our company holds dear. Our team of people really do value, amongst all other things, above all other things, patients first. The work that we do, whether it be in manufacturing, in designing and conducting toxicology studies and clinical trials and so on, is really all about thinking about how do we develop these products for the benefit of our patients safely and ethically. The other values that we hold dear relate to integrity, respect, high levels of performance, being innovative in what we do, being accountable to each other and our shareholders, and being excellent at what we do. And that's something that we all try and, these are all values that we try and live out in our interactions with each other and with those outside of the company who we interact with. These are the people who deliver those values and deliver on the programs that Amplia is working on. I couldn't be more proud of being able to work with this group of people and having assembled such a high caliber group of excellent people who are incredibly good at their jobs and are incredibly professional as they go about their work. It's a real delight working with this team of people. If I go over the highlights for our achievements during this financial year, it really has been a year of delivering on the things that we promised to deliver on for our shareholders. We've established the AMP945 clinical trial in patients with pancreatic cancer, and we initiated recruitment of that recently. We said we were going to conduct toxicology studies that would allow us to assess AMP945 for extended periods of time in patients with pulmonary fibrosis, and also will support our other submissions, regulatory submissions down the track. Those studies are nearing completion and we're starting to see initial results. We've been working on improvement of AMP945 manufacturing methods in readiness for commercialisation. At the end of the day, if AMP945 is successful in the clinical studies that it's being tested in, we need to be able to actually manufacture a commercial product that can be sold through the pharmacy. And so a lot of that work has been going on in the background as well. We interacted with the United States Food and Drug Administration in order to validate or check in with them on our development plans for AMP945, and the feedback we had from that meeting was very positive. We've also been spending time on surveying the opportunity landscape for AMP945 and AMP886. Are there other indications or sub-indications that we should be developing these assets in? Working closely with my colleague and Chief Operating Officer, Rhiannon Jones, We've built the right team around us to deliver on the company's objectives. And in doing that, we've brought in people not only with the right skills, but also with the right approach and the right culture. The company finished the financial year in a very strong cash position, holding $14.6 million in cash at the end of March 2022. You may have noted in our recent Appendix 4C statement that cash at the end of July was sitting at $12.7 million. The burn rate for this cash will be substantially driven by our rate of recruitment into the ACCENT trial. So things such as investigator fees and pass-through costs get paid as patients accrue into the trial rather than before. We're also expecting R&D tax rebates from the Australian federal government to provide income based on our expenditure in financial year 2022 and financial year 2023. So we feel as though the company is currently in a very strong cash position. One of the things I'm particularly focused on and proud of is Amplia's capital efficiency. In the financial year ending in March 2022, our expenditure in total was $5.63 million. And 2/3 of that was directed towards developing our assets and increasing their value. We obviously recognise that there needs to be expenditure on salaries and superannuation, and there are corporate costs associated with being a publicly listed company. But really, the way we've set Amplia up, we direct as much as possible into development of our assets. And compared to other companies of our ilk, I think that Amplia is an incredibly capital efficient company. One of the reasons for this is the way we conduct our clinical development activities. We run a very sort of virtual model where development managers within the company coordinate the activities of various experts outside the company whose services we can turn on and off as needed. And this is an incredibly efficient way to run a biotech company, but it's also a way that allows us to access experts who really have been down these paths before. And we don't need to reinvent and rediscover key elements of developing oncology and fibrosis assets, because we're tapped into experts who've been down this path. There are numerous achievements and outcomes that the company has been able to deliver during the last year. A lot of these have been focused on the initiation of the ACCENT trial and getting through various steps leading to that. So things like manufacture of more drug substance on a 2 kilogram scale earlier on in the year, going through ethics approvals, opening the first site and recruiting the first patient are all things that we've been able to achieve throughout the year. We've also done some really nice work looking at pancreatic cancer models and revalidating, I suppose, our approach to pancreatic cancer. We presented at a major international conference, the American Association Cancer Researchers conference in April 2022. And we're about to present again at another AACR conference, which is more focused specifically on pancreatic cancer. So it's been a very busy and productive year for the company, where we've essentially taken a phase 1 asset through to the initiation of phase 2. So just to talk about our technology, Amplia Therapeutics is developing inhibitors of a target called focal adhesion kinase, or FAK. Our inhibitors are small molecules, they're orally bioavailable, and they're incredibly potent and selective for their target. We're developing these in 2 main therapeutic areas, namely oncology and fibrosis. We're at the clinical stage of development with these, with our most advanced clinical trial being in first-line patients with advanced pancreatic cancer. And we have another clinical trial in pulmonary fibrosis in the planning stages. FAK, or focal adhesion kinase, plays a central role in developing and supporting essentially the growth of fibrotic tissue. So on the left of this slide, FAK plays a role in laying down collagen, which is one of the most important building blocks for fibrotic tissue. And it also supports cross-linking, or I guess roping of that collagen into tight-knit bundles, essentially building the tissue that forms the tough scar-like tissue that you'll be familiar with. In oncology, focal adhesion kinase also plays a role in supporting fibrosis and building up tough fibrotic or scar tissues around many solid tumours. It also plays a more subtle role in immunosuppression, where focal adhesion kinase leads to suppression of the body's normal ability to reject solid tumours in the tumour microenvironment. So the company's pipeline looks like this. AMP945 is being developed in pancreatic cancer as a combination therapy, and it's around about the bridge between phase 1B and 2A at the moment. 945 is also being tested we also plan, I should say, to test AMP945 as a monotherapy in people with pulmonary fibrosis. We're also looking at AMP945 in other oncology indications in preclinical studies, as well as AMP886, another FAK inhibitor, in combination studies. In oncology, this is our hypothesis. Fibrotic tissues essentially shield and protect many solid tumours from chemotherapy. We believe that our FAK inhibitors may have the ability to remove that shield, and once unshielded, those tumours will be more susceptible to chemotherapy. So the story in oncology is that we're all about trying to improve or enhance or amplify the effectiveness of chemotherapy in patients with pancreatic cancer. The evidence for this comes in some of the work that we've been doing primarily with the Garvin research group, where we have shown, as I'm showing on this slide, in two different pancreatic cancer models, the so-called KPC and the TKCC models, that when you add AMP945 to gemcitabine and Abraxane, or nab-paclitaxel, which is a common chemotherapy used to treat patients with pancreatic cancer, AMP945 appears to increase the survival of mice, whether they're implanted with the KPC model or the TKCC model. You'll notice that the TKCC model is appears to be a lot longer than the KPC model. That's because the KPC model is a very aggressive model, and the TKCC is a slower growing model, where the pancreatic cancer cells are actually derived from a real human cancer. So in both these experiments, AMP945 appears to enhance the effectiveness of gemcitabine and nab-paclitaxel. Our hypothesis in pulmonary fibrosis is that using a FAK inhibitor will slow the rate of the development of fibrotic tissue in the lungs. So we know that idiopathic pulmonary fibrosis is a really dangerous progressive disease, and the current treatment options for it are very poor. FAK plays a role, we know, in the establishment of that fibrotic tissue. And our preclinical models, again, show that AMP945 is able to reduce lung fibrosis. The safety profile of AMP945 so far appears to be excellent, which supports its potential use in long-term chronic therapy in these patients. So taking together the safety profile of AMP945 and the apparent efficacy of it in these preclinical models of pulmonary fibrosis, we believe that we have a rationale for conducting a clinical study in these patients. This is some of the evidence that AMP945 is an antifibrotic agent. So going left to right on this slide, we're looking at increasing concentrations of AMP945 in an in vitro experiment where we're looking at the ability of cells that produce the building blocks of fibrosis to produce those building blocks. And you can see that this pink stain, which stains those building blocks or collagen molecules, is more intense over here where the concentration of AMP945 than it is over here where the concentration of AMP945 is higher. So we call this a dose response, which means that as we increase the dose of AMP945, we see depletion of collagen in this case. We also are able to see in the same sort of experiment, going left to right, increasing AMP945, that AMP945 limits cross-linking of collagen, which you can think of as essentially decreasing the ability of these collagen fibers to form themselves into tough strands. So now I'll move on to development of AMP945 and the ACCENT clinical trial. This clinical trial is a study of AMP945 in combination with gemcitabine and Abraxane in people with advanced pancreatic cancer who haven't been treated before. It's a single-arm, open-label trial. So as patients are recruited into this trial, we will know what they are being treated with. These patients, as I indicated, need to have been previously untreated with gemcitabine and nab-paclitaxel, and the primary endpoint of the trial is the objective response. So in other words, in this trial, we will be measuring the size of tumours and determining whether or not those tumours shrink relative to their size at the start of the trial. While the trial is conducted under a single protocol, there are 3 stages to the trial. Part A is a dose confirmation, where we will recruit around about 12 patients, and we will gradually escalate the dose in those patients up to the point at which we think we have optimized that dose. Once we have optimized that dose, we will take that dose and combine it with nab-paclitaxel and gemcitabine in around about 50 patients, and those patients will be recruited in 2 stages. Initially, in an exploratory efficacy analysis, we will see whether or not AMP945 is likely to be improving, or at least not making treatment with gemcitabine and nab-paclitaxel worse. Subject to the success of that stage, we will move on to stage 2, where we will verify the efficacy of that combination. You can find more information about this clinical trial by essentially punching this number into a web browser, and you will find the clinicaltrials.gov registry entry for this trial. A lot of our work during 2022 has been on setting up this AMP945 trial in pancreatic cancer patients. And there are a number of stages that need to be gone through in order to achieve that. First of all, we needed to, having completed writing the protocol, we needed to seek ethics committee approval from 2 different ethics committees in this case, one in New South Wales and one in Victoria. And we obtained those ethics approvals respectively in April and May of 2022. The next stage in the process is contract negotiations with the individual sites who are going to conduct those clinical trials, who are actually going to recruit patients. Having finalised the contracts, it's a very important part of the process to conduct a site initiation visit where essentially Amplia goes to the site, either physically or virtually these days, and trains the site in the protocol, what the background science is, and brings them up to speed with a lot of nuances, I suppose, of the clinical trial. Once that site initiation visit has been completed, we can ship drug to the site and initiation of recruitment can begin. During our previous disclosures, Amplia forecast that we were planning to initiate recruitment in the first quarter or second quarter of 2022. This didn't occur, primarily because the ethics approvals and the site contract discussions at the beginning of the process above were slowed down significantly, largely due to staffing shortages in the hospitals with which we were discussing. And of course, this would have a knock-on effect to site initiation visits and recruitment. So in the end, we were able to recruit our first patient in August 2022, roughly 1 to 2 quarters behind our initial forecast. From here, then, we've taken a number of steps, or tactics, I suppose, to optimise recruitment running down the track and make up for any time that has been lost. We're adding more sites into the Australian portion of the trial in order to minimise the risk of there being any recruitment bottlenecks in parts A and B of the trial. You may have noticed a recent media campaign that we ran through mainstream media on Channel 9 and in the Australian newspaper. And that media campaign has done a lot to raise awareness about the trial. We've had a lot of inquiries from patients and their doctors and patient advocacy groups since that publicity. And I'm sure that that has helped raise a lot of awareness about the trial. We're also working on initiating Korean sites. Korea was always part of our plan for Part B of the trial, and we are now initiating those discussions with Korean sites because they have a very good reputation as high volume, if you like, recruiters in pancreatic cancer clinical studies. So in the next 3 slides, I'd like to talk you through a little bit about the strategy and the process that we have implemented for the execution of the phase of this clinical study. Over on the left of this slide discusses our regulatory and recruitment strategies for the trial. So the regulatory elements that we've been conducting, first of all, are the ethics committees approval and the clinical trial notifications for Australia. These are required steps in order to initiate recruitment in this country. We've also conducted pre-IND interaction, as I mentioned at the beginning of this presentation, with the United States FDA. And that's an important part of our regulatory strategy because it provides not exactly certainty, but some form of validation that the clinical protocol that we are about to execute is of an acceptable nature to the US FDA. We've started Part A dose selection, so that's underway, and our aim here is in Australia to recruit across 4 to 8 sites about 12 patients. While recruitment for this Part A is going on, we are also initiating our discussions with the Korean regulatory agency and preparing a Korean investigational new drug application. So rather than sitting back and waiting for this Part A to complete, we're putting the pieces in place for what needs to come down the track. We're also preparing a IND for the United States FDA. But our approach here is that we do not plan to file that IND until we've selected the dose from the Part A part of the trial. The reason we do that is because we think it would be better to go to the United States FDA with the proposed dose that we plan to use in Part B of the trial and explain to them the data that we've received and the rationale for selecting that dose and getting their input relatively early in the process. Having selected that dose, we will run sites, both the same sites in Australia, and we will open up new sites in South Korea, subject to the Korean regulatory authorities authorising us to do so. And then we will recruit about 26 patients across the Korean and Australian sites. After that, we'll conduct our interim analysis, and if that interim analysis indicates that the trial should be continued, then we will continue recruiting sites at the patients at the Australian sites, the Korean sites, and for stage 2 of Part B, we'll bring in US sites, assuming that we have opened an IND successfully back here through the US FDA. So it's a very sort of staged approach where we interact with the regulatory agencies as we need to in order to initiate studies, but also, particularly in the case of the US FDA, to get their feedback before we open sites in their country of jurisdiction. In terms of timelines, we're expecting Part A of the trial, the dose selection portion, to take about 9 months. The 26 patients that we will recruit into the Part B stage 1, we expect to take about 10 months before we are able to conduct our interim analysis. Part B, as recruitment rate per month, is expected to be higher than Part A because we're not going through the same level of safety assessment before we do a dose escalation. So Part B recruitment should be more rapid in terms of patient per month per site than Part A. What this slide shows is that there are multiple decision points as we conduct this trial. In Part A, where we are at the moment, there's a number of decisions. At each of these dose escalations, should we escalate the dose or not? Is it safe to do so? Are we likely to obtain a greater level of efficacy if we do escalate the dose? So we're making a number of decisions as we go through Part A. At the end of Part A, we have to look back on the data that we've collected here and say, okay, what is the optimal dose? What is the dose that we will take into Part B? And that's another key decision in this trial. The next decision is, have we found, when have we found in Part B more than 5 responders? If we find more than 5 responders in this group of 26 patients, then the decision to then we make a decision that we would proceed with Part B of the trial. If we do not find more than 5 responders in this part of the trial, then we would conduct this detailed interim analysis, and we would decide whether to proceed into Part B or not. And at the end of the trial, the decision is whether or not we have found more than 15 responders in our now 50 or so patients. If we have, then we proceed with further development of AMP945. So there are multiple decisions that we're making as we conduct this trial, together with the work that we're doing with the regulatory agencies in order to be ready for the different stages of the trial. The sites that we are currently working with in Australia are shown on this slide here. So Western Health and Monash Medical Centre, beg your pardon. Western Health and Monash Medical Centre are sites open for recruitment. Westmead and Royal North Shore Hospital in Sydney are close to recruitment, as are the Epworth in Melbourne. We have follow-on sites that are serving both as a contingency for Part A and for other sites that may be poor recruiters in South Australia, another one in the Sydney area, and 2 in the Brisbane area. These sites have been selected on the basis that we would not need further ethics approval to bring those sites into the trial, we would simply need to negotiate contracts and initiate those sites, should they be needed. In this slide, what I'd like to do is just focus in a little bit on more detail around the part A of the trial, the part of the trial that's running at the moment, and provide you with a little bit more detail about how this works. So we're now at dose level 1, where we're seeking to recruit 3 patients. Once we recruit those 3 patients, at the end of their first cycle of chemotherapy, we'll look at their safety, whether there's a pharmacodynamic effect, whether we're hitting, that is to say, whether we are hitting FAC in those patients. We'll also conduct a pharmacokinetic review. I should mention that the dose we are using in this group of patients here was the highest dose that we achieved in the study in healthy volunteers conducted last year, and was also a dose at which we showed that there was a pharmacodynamic effect. So we're expecting to see a pharmacodynamic effect in this group of patients as well. But what we don't know about is what the effect of combining AMP945 with chemotherapy will be. So that's what we're doing there. Assuming we see all these things and it's safe to proceed, we will escalate the dose in another 3 patients, so 3 different patients now, and we will conduct the same safety and pharmacokinetic and pharmacodynamic analyses. Likewise, again, assuming everything is okay here, we will escalate the dose again, and so on up to dose level 4. This dose escalation will stop if we see either a saturation of the pharmacodynamic effect. In other words, we see no enhancement of efficacy or pharmacodynamic or interaction with FAC if we dose escalate, in which case there's no need to escalate the dose. It will also stop if we hit a safety ceiling. In other words, if for some reason patients can't tolerate the increased dose of AMP945, then clearly we would not dose escalate beyond that point. So what the outcomes of this part of the trial will be an understanding of the safety and tolerability in this population of AMP945 who are also receiving chemotherapy for their illness. We'll be looking at efficacy in this trial, of course. We don't know whether that efficacy will be optimal or not, but we will certainly be looking at the objective response of these patients to the addition of AMP945 to their chemotherapy. And I should stress that this is an open label trial, so we know what these patients are receiving and what dose. So there are no blinding issues that we need to concern ourselves with. Finally, the outcome of this part of the trial will be selection of a dose, we hope, for conduct of Part B of the trial. So just to summarise a few of our plans for the financial year 2023, for the year coming. We expect there to be significant clinical milestones during the year. As I just indicated, we'll be looking at the efficacy data as it emerges from this ACCENT trial, as well as the safety data. We're also planning to initiate a pulmonary fibrosis clinical trial sometime during the coming year. We'll be engaging with the regulatory agencies at the US and Korean regulatory agencies through the INDs, and we'll be obtaining feedback from both those agencies. We're also always looking to expand the therapeutic opportunities for AMP945 and AMP886 in preclinical experiments. So I think the promising year, the coming year, is going to be an exciting one for Amplia. There are a lot of promising things going on in the company, both at the preclinical and at the clinical stage. And I'm looking forward to keeping you updated. Thank you very much for your attention today, and thank you for dialing in.